Hello everyone, myself is Deepika. In this video, we are going to see about cDNA transformation. cDNA is nothing but a double stranded DNA that's synthesized from a single stranded RNA template. In a reaction catalyst, we use reverse transcriptase as an enzyme. So, once cDNA is created, that is from a mature mRNA, means it's originated from a eukaryotic cells with the use of reverse transcriptase enzymes. In eukaryotes, a poly-A tail consists of a long sequence of adenine nucleotides facilitates to distinguish the mRNA from mRNA from tRNA and rRNA. Therefore, they are used as a primer site for the reverse transcriptase. So, this is the process of synthesizing of cDNA. Let's see about construction of cDNA. There are six types: isolation of mRNA, synthesis of first strand of cDNA, synthesis of second strand of cDNA, cloning of cDNA, introduction to host cells, clone sections. For this picture, we are going to see step by step how we can get cDNA. First, isolation of mRNA. We can extract a tissue with a gene of interest is prepared and extract free from that extract must be free from proteins, polysaccharides and all other contaminants. So technique use of dioxyamide cellulose chromatography is used for the further purification of many eukaryotic mRNAs from the total or polysomal fractions. mRNA consists of poly A, that is adenosine residase tails at their 3 dash end. There is a 5 dash and 3 dash. For cDNA, the reverse transcriptase is acquired, in which 3 dash to 5 dash will be acquired. Under favorable condition, this tail will be bind to a string of thymine residents immobilized on cellulose and the poly A plus fraction can be elected. Two or three passages of a poly A fraction through such as column produces a fraction highly enriched for mRNA. This fraction includes a different mRNA sequence. However, certain techniques can be employed for extracting a particular mRNA species. After the preparation of the fraction, it's essential to confirm its extracted mRNA consists of the sequence of interest. Then synthesis of first strand of cDNA. So reverse transcriptase is a RNA dependent DNA polymerase that is used to copy the mRNA fraction into a first strand of DNA. If enzymes like other DNA polymerase that can also add residents of a 3-OH group of a existing primer, which is a base pair with the template. Most commonly, a primer is used for is oligo-TT for cloning of cDNAs. Oligo-TT primer is 12. It has 12 to 18 nucleotides in length that binds with the poly A track at the 3 dash end of the mRNA molecule. The RNA strand of a resulting RNA DNA hybrid is destroyed primer to second strand synthesis through alkaline hydrolysis. Let's see about a synthesis of a second strand of cDNA. In second strand of cDNA, it can be synthesized by two techniques. There are self-priming cDNA and replacement synthesis. First is we see about self-priming cDNA. Self-priming cDNA is nothing but it's mRNA that a hybrid and degenerated for the synthesis of second strand of a single strand of cDNA by the null fragment of DNA polymerase 1. If the transitory hairpin is a structure that present at a 3' end of single stranded DNA that can be used to primer and synthesis of second strand of cDNA by null fragment of Escherichia coli DNA in polymerase 1. If a single stranded specific S1 nuclease digests the hairpin loop and any single stranded overhang at the other end, ultimate product is a population of double stranded blended DNA molecules complementary to the original mRNA fraction. Then let's see about replacement synthesis. In this method, a cDNA, that is mRNA hybrid, works as a template for a nick translation reaction. In the mRNA strand of hybrid, RNase H produces nicks and gaps, creating a series of RNA primers. These RNA primers are used for Escherichia coli DNA polymerase 1 during the synthesis of second strand of cDNA. What are the advantages by using this technique? Means it's a very effective. It can be performed directly using a product of the first standard reaction. It eliminates to need to use nuclease S1 to cleave the single stranded hairpin loop into a double stranded cDNA. Next, cloning of cDNA. It's a most frequently used technique for cloning CNAs involves the addition of complementary homopolymorphic tracks to double-stranded cDNAs and to the plasmid vector. To the cDNA, string of cytosine residents are added using the enzyme terminal terferase to form oligo-DC tails of the 3' ends. Likewise, a plasmid is cut open at a unique restriction endonuclease site and tailed with oligo-DG. Now the vector and a double-stranded cDNA are joined by hydrogen bonding between the complementary homopolymers. In the results, the formation of open circular hybrid molecules capable of transforming E. coli. Introduction to host cells. For the transformation of bacteria, the recombinant plasmids are used usually in E. coli K12 strains. This is uptake of plasmid molecules from the surrounding medium is performed by E. coli cells treated with calcium chloride. If any gaps in the recombinant plasmid, that will be repaired by the host cells. The transformed bacteria can be isolated from non-transformed one on the basis of antibiotic resistance. Majority of cloning plasmids contain two antibiotic resistance genes, one of which is destroyed during cloning. For instance, in the case of PBR322 cloning into unico-PSTL site destroys ampicillin resistance but leaves tetracycline resistance intact. Bacteria transformed with a recombinant plasmid will be sensitive to ampicillin but resistant to tetracycline. Next, cloning section. The cloning process generally commences with the whole population of mRNA sequence. If a selection of clone carrying the sequence of interest, it's a tough job. If gene is expressed, then the simplest selection is to screen for the presence of the protein. It can be screened either by practical phenotype to produce or by the protein reduction method using based on immunoglobulin immunology or enzymology techniques. If the protein is not expressed, then other methods such as nucleic acid hybridizations are used. Identification of the gene is discussed after the genomic DNA cloning. Transformation. Transformation means if a cDNA transformation is a technique that used to introduce a cDNA molecule into host cells. This is often done to express specific gene or study gene function in the host organism. Transformation can be accomplished using various methods such as electroporation, chemical methods, or viral vectors. So, transformation methods there are three types: bacterial transformation, yeast transformation, and mammalian cell transformation. First, we see about bacterial transformation. In bacterial transformation, the cDNA is typically introduced into E. coli cells. That cells are made competent as able to take up DNA through chemical treatment or electroporation. Complete cells are mixed with cDNA and subjected to heat stroke or electroporation to facilitate DNA uptake. Transformed cell containing cDNA are selected using antibiotic resistance makeover on the vectors. This is how we can add bacterial transformation. Let's see about yeast transformation. Yeast cells can be transformed with cDNA by using various methods such as lithium acetate treatment or electrophoresis. Selection markers, that is auxotrophic markers on the vector, help to identify yeast cells that can take up the cDNA. Next, mammalian cell transformation. Transfecting cDNA into mammalian cell that can be achieved using methods like calcium phosphate precipitation, electroporation, or liquid-based transfection reagents. Cells are cultured under a suitable condition of cDNA expression. So this is a overall methods we can use for collecting and getting the cDNA. So conclusion. cDNA is a transformation is a fundamental technique that is used in a molecular biology, allowing researchers to manipulate gene expression and study gene function in various host organisms. The choice of host or vector and transformation methods should be tailored to the specific research objectives. Thank you all.